Hello everyone, this is part 2 of Unit 7 UGC NET JRF Forensic Science, in which we will discuss about pollens and diatoms. The study of pollen grains is known as forensic palynology, and the presence of pollens on clothing, earwax proves the normal habitat or natural habitat of the person. So, we can find out from where the person belongs using the pollen grains. Pollen grains are abundant in almost all environments. They are very durable and persist on surfaces for many years. So these are the characteristics that make pollen a very good trace evidence. And it is based upon the Locard's exchange principle that every contact leaves traces. So whenever a person comes in contact with the pollen, it gets transferred to its shoes, to its clothing, hair, etc. The hard outer layer of pollen grain is made up of sporopollenin, and this sporopollenin may make it resistant. It is the hard layer, and due to this layer, pollen grain is very durable. As we all have studied, pollen grains are male reproductive units of plant, and these are produced in flowers, male flowers, or the bisexual ones. The pollens produced by flowering plant, conifers, the spores produced by ferns, all these are microscopic and not visible to the naked eye, so that the suspect or the criminal avoid them. But these microscopic creatures help in identifying the habitat of the suspect, from where it belongs. The morphology of the pollen grain determines the place of the offense, whether the, the suspect is related to the crime scene or not, etc. Pollen grain study can also tell us the time of the year in which the crime took place, as some pollens are released in a particular seasons only. Moving on to diatoms. So diatoms are a group of eukaryotic algae and the most common type of phytoplankton. These are unicellular and sometimes may form chains or colonies. The characteristic feature of diatom cell is the unique cell wall made up of silicate. So, also after a long period of time, the cell wall of diatom secretes substances and it forms diatomaceous earth. The presence of diatoms in lungs or other body tissues uh, help us to determine whether the death is due to drowning or not. Diatoms are present in both lotic and lentic water bodies. Examination of diatoms. In drowning cases, identification of diatoms in the bone marrow is done, and the type of the species of diatom present in the water body is compared with the one present in the bone marrow. Now the diatom extraction methods. First is direct digestion of material using nitric and sulfuric acid. Then enzymatic digestion using proteinase K. In case of indirect digestion, there will be incinerate in electric oven, charring, dissolve in corrosive acids. Now, first collect the diatoms from the water body, treat them with acid. Here we use concentrated HCl. The supernatant is discarded. Then add concentrated sulfuric acid, which will turn the material into blackish color due to charring of organic material. Supernatant was cooled and then solid sodium nitrite is added. The mixture is reheated and it turns brown, and then finally a clear solution is obtained. Distilled water is used for the washing, and the residue was resuspended in acetone. So this method is known as acid digestion method. The characteristic features of diatoms: there will be presence of photosynthetic pigments, chlorophyll A and C, xanthophyll and fucoxanthin. The silica cell wall of diatom is composed of two walls or shells. It is like a soap box. So the outer part is known as outer epitheca. Outer part, or you can say outer wall, is epitheca, and inner part or inner wall is hypotheca. And based on the pattern found on these cell walls, they are classified as centrales and pennials. In centrales, marking are arranged with reference to a central point. In pennials, there will be a structural or primary element, and the secondary structures would be arranged according to it. Diatoms are highly polymorphic on the basis of their shape and size, and the study of diatoms in water is known as limnology. In drowning cases, diatoms enter the bloodstream through lungs and are carried towards the brain, liver, bone marrow, etc. Bone marrow removed from the femur is the most popular choice for examination, is the most popular evidence for in drowning cases, as it is indicative of ante-mortem drowning, as only if the person is alive, so the blood will flow and will reach the bone marrow. Diatoms can reveal the place of drowning, and in highly putrefied bodies where other signs are not there, it is helpful in establishing the cause of the death. So in case of drowning, the most common evidence found is diatoms. Thank you for watching. Stay tuned for more such videos.